So today we will be presenting on the synthesis of amino triester lipids and their applications as biodegradable surfactants for drug delivery and precise control of quantum dot formation. In this project, we took on a broad spectrum of chemistry, including chemical synthesis, biochemistry, and a little bit of physics. First, we'll be going over a bit of background surrounding lipids, including physical properties and history of using lipid nanoparticles in drug delivery. Then we'll go over a bit of organic chemistry, talking about our synthesis of tripod lipids and characterization of a ladle high-field NMR. After that, we'll discuss how we used in vitro cell proliferation assays and fluorescence imaging to biologically evaluate their properties. And lastly, for a bit of inorganic chemistry, we'll talk about our synthesis of quantum dots. So to start off, we want to explain some physical properties of lipids. And as we know, butter is a solid and olive oil is a liquid. So why is that? And this is due to the difference between saturated and unsaturated fats. If you didn't know, a saturated fat is a lipid that does not have any present double bonds or alkenes on the hydrocarbon chain, and therefore can compact really nicely together to form a solid. Uh, Unsaturated fats, on the other hand, have alkenes present on the hydrocarbon, which allow it to have fluidity or bending. There are different types of unsaturated lipids, such as monounsaturated lipids, polyunsaturated lipids, with which each have one or two or more lipids on them, alkenes on them respectively, as well as cis versus trans unsaturated lipids, which have either hydrogens on the same side or on different sides. Lipids can also form a lipid bilayer, which will have a polar hydrophilic head on the outside and a nonpolar hydrophobic tail on the inside.
Okay, so one of the problems with the lipid bilayer that Anna just mentioned is that it could be hard for certain drugs or oligonucleotides to get through, even if we want them to for medicinal purposes. For example, nucleic acids are polyanionic materials that themselves exhibit poor membrane permeability. So this means that they have high polarity and charge, which is typically caused by the phosphate backbone's negative charge, and a large molecular size, as RNA is made of many different nucleic acids. One possible solution to this problem is to use lipid nanoparticles, which we refer to as LNPs. Their positive charge can help.
I'm gonna increase your gain real quick. We can't hear you. Okay, there we go.
Should I restart?
No, just keep on talking.
Okay. Um, sorry. One possible solution is to use lipid nanoparticles, which we refer to as LNPs, because their positive charge can help neutralize these anionic nucleic acids and help them get through the membrane. So in the bottom right corner, you can see a pie chart showing the molar ratio of lipids that are used for LNP formation. This shows that ionizable lipids, which we chose to use in our study, are already really common in medicinal chemistry. So two examples of drugs that make use of this lipid nanoparticle RNA delivery system are the COVID-19 drugs developed by Moderna and Pfizer. So if you note the similarities of these two drugs, first of all, they both have a polar ionizable head, and they also have the kinked or branched lipid tail. The good part about these lipid tails is that they remain neutral at normal pH levels, which means that they are not really toxic and won't have off-target toxicity. However, in the acidic endosome, they can become positively charged. This helps them better bind to and package RNA. Now, the example of how alkenes influence these properties can be found in the study reported by the Kim lab in 2016. So you should note that the only difference between these two structures, DSPC and DOPC, is the presence of an alkene in DOPC, whereas DSPC consists of fully saturated lipid chains. However, by just changing this one small part of the molecule, they were able to observe significant biophysical differences. So if you take a look at this chart in the upper left corner, in their study, they realized that if they were to increase the amount of DSPC lipids compared to DOPC lipids, they were able to change the gel-like phase into a completely liquid crystalline phase. In addition, membrane thickness also increased as they added more DSPC. So we decided to take inspiration from these compounds and use this oleic acid and stearic acid structures as part of our tripod lipids. So aside from the drug discovery road, tripod lipids are also used in material science, and they can typically be used for softening purposes, which means they can be found in shaving cream, laundry detergent, or even toilet paper. So next we will be covering how we are synthesizing our tripod lipids. The first thing to note is that all of these conditions are non-hazardous and pretty much mild synthetic conditions. We use a mechanism called EDC coupling, and this reaction is completed in roughly 18 hours, so we just let it run overnight. Starting with stearoyl in red, this is our most saturated lipid, meaning it has no double bonds and just straight carbon chains. As expected, it's the most solid of all our lipids and appears as a waxy substance at room temperature. Next is oleo, which is monosaturated, meaning it has one alkene and has a cis double bond. The kink in the structure gives it a much more fluid character. It was visibly liquid-like during handling and synthesis, even at room temperature. Lastly is the linaloyl tripod lipid, which is the only polyunsaturated lipid that we are using. It has two cis double bonds, and the structure is even more kink, giving it an even more fluid-like character. So when we're handling this compound, it appears as an oil. Next, we have elatic. Our elato tripod lipid is mono-unsaturated, but it has a trans double bond instead of a cis double bond. Because the hydrogens are on opposite sides of the double bond, the chain stays like fairly straight, so the molecule still behaves like a saturated fat. We observed it as a firm wax, not really like a liquid. Lastly, we did try to synthesize a fifth compound using a lipoic acid. This contains a 1,2-diethylene ring rather than just a long lipid chain. However, this reaction ended up having really messy converging, so we abandoned it early on in the process. So the way that we are figuring out if we actually made the right lipids is through an analytical technique called nuclear magnetic resonance. So above is our spectra for stearoyl, oleo, and linaloyl tripod lipids. And if you don't understand how to read these graphs, don't worry, because we're about to explain how it works. So in order to analyze the NMRs, you have to first understand the theory behind it. So essentially, when you're putting your compound into your NMR tube and then inserting it into the NMR machine, you're putting it into a magnetic field. Within this magnetic field, the nuclei of your compound can distribute into two different energy levels. And some are, that's the alpha spin state and the beta spin state. The alpha spin state is lower in energy since the nuclei is oriented with the applied field, whereas in the beta spin state, it's higher in energy because it's opposed to the magnetic field. So in the machine, the electromagnetic radiation matching the energy difference is applied, and protons are able to absorb that energy, matching the gap between your alpha and beta spin states. This causes them to flip, so all your nuclei flip to the beta spin state. As the protons relax back, they release the energy, and this released energy can then be converted to the ppm value that you see on the spectra. So as I stated, since the ppm value depends on the alpha and beta spin states, it also depends on how much energy is actually received from the magnetic field. And this is influenced by what atoms the proton is bonded into. So if the proton is next to a really electronegative atoms such as oxygen, nitrogen, or fluorine, that atom is able to pull electron density away from the hydrogen. That makes the hydrogen less shielded, meaning it feels more of the external magnetic field. And as a result, it absorbs a higher energy in order to flip the spin states, giving it a higher ppm value. So in this example, you can see the electronegative element, oxygen, is highlighted in red. And since that's able to pull the electron density away from its neighboring carbon, the carbon highlighted in blue would be further downfield as its electron, or as it is more deshielded. The purple carbon, which is further from the electronegative element, will appear further upfield as it is slightly more shielded. So now that we know how an NMR works, we'll be going through how we're actually using that information to characterize our allyl high field NMR. So starting with the peak around 5.5 ppm, usually it'd be really helpful while you're analyzing your NMRs to have this 1H peak shift chart to look at. Essentially, when you see the shift is around 5.5 and you look at the chart, you can realize that usually it's alkenes that have that chemical shift. Since we do have alkenes on our tripod lipid, we're able to assign that to the specific peak. Next would be the peaks highlighted in pink and bright blue. And these are close to the oxygen, which does withdraw electrons. And it's worthy to note that the bright blue peak is corresponding to the carbon next to the tertiary amine. And the amine is also a electron withdrawing group, just not as strong as the oxygen, which is why it's further upfield. Next is the orange and green peaks. These are also impacted by the ester. Now, you'll see that I skipped a peak between those two. And this one we integrate differently because when you open up the integration, which is the area underneath the peaks, you'll realize that it actually integrates to double of all the previous peaks we've been looking at. So this means that it must be next to the alkenes as they are all in the similar environment. Next, we have the methyl group. This group is the furthest upfield because it has the least electron withdrawing forces nearby, as it is only connected to one other carbon. In addition, you can also cross-reference that with any NMR shift graph you can find online. And as you can see, a methyl group, which is an alkyl CH group, should be really far upfield. So lastly is our lipid chain, which is all the other ones that we haven't accounted for so far. And since we have 10 carbons unaccounted for on each chain so far and 3 chains, it should integrate to around 60. And yeah.
In order to probe the efficacy of our lipids in delivering compounds across the cell membrane, we needed a high-throughput method to quickly screen for the effects of lipid co-delivery. We decided the best way was to conduct a series of cell proliferation assays tracking cell viability and membrane degradation when cancer cells are dosed with a mixture of our lipids formulated with FDA-approved anti-cancer drugs. Through a preliminary MTT and LDH assay, it is shown that our lipids are non-toxic against the metastatic breast cancer cell line MDA-MB231, and they did not induce cell lysis, meaning that our lipids may have potential as structural components of larger drug delivery systems. MTT and LDH assays are in vitro assays that provide information on the amount of living and dead cells in a population of cells that have been drugged with the compound. This information is displayed through an optical readout, often where a yellow tetrazolium salt is reduced into a formazine dye that can be detected at specific absorbance wavelengths. The MTT assay evaluates cell viability based on the metabolic activity, where the reagent MTT thiazole blue tetrazolium bromide, is added to your 96-well plate after your cells have incubated in drug media for a desired amount of time. The plate is then put back in the incubator, and over time, the yellow MTT is metabolized into purple formazan, which absorbs light at 570 nanometers. After you see the purple crystals accumulate, you can then remove all the media, apply DMSO, which will pop all the cells, and the purple formazan crystals will be dispersed. You can then scan the plates with the plate reader to quantify how purple the samples are, which tells you how many cells are alive, or were alive. This allows you to calculate what percentage of cells were metabolically active, which correlates to the percentage of cell viability, since only the living cells will metabolize the MTT into the formazan. Next slide. The LDH assay, or lactate dehydrogenase assay, evaluates the cytotoxicity of a drug based on the amount of LDH released, where LDH is a stable cytoplasmic enzyme found in all cells and is released into the cell culture supernatant upon damage to the cell membrane. We measured the reduction of a yellow tetrazolium salt, INT, by NADH into a red, water-soluble formazan dye by the absorbance at 490 nanometers. The amount of formazan is directly proportional to the amount of LDH in the culture, which in turn is directly proportional to the number of dead or damaged cells. After incubation with drug media, some of the supernatant is removed and placed into separate flat bottom plates. Tritonix, a lysine surfactant, is then added to select wells where there are no drugs to act as a positive control. An LDH solution is premixed and added into the supernatant media, and the plates are incubated. The Tritonix controls will be the darkest red in color, and one can then calculate the percentage of LDH released by scanning the plate through a plate reader. So we ran a series of MTT and LDH assays on 3 cell lines. We first prepared dilutions of the 6 drugs shown on the center of the screen, which are all FDA-approved anti-cancer therapeutics that are known to reduce cell viability. We made a concentration gradient through serial dilution, and we mixed our tripod lipids with the drug dilutions in equivalent volumes inside of a V-bottom plate. The mixed solution was then added to cell culture media, which was then applied onto cancer cells seated in 96-well plates. Next slide. Yeah. We drugged the human colorectal cancer cell line HCT116 with our 6 selected anti-cancer therapeutics, and we ran an LDH assay to observe the percentage of LDH released when the drugs were co-delivered with our tripod lipids. As you can see, the percentage of LDH release in the presence of lipids generally did not increase much in comparison to the no lipid control. Furthermore, we co-delivered our lipids in 2 additional cell lines. The first is HT29, which is a human colorectal cancer cell line, and the second is A549, a human non-small cell lung cancer cell line. We normalized and calculated IC50 values for all the drugs we delivered for every lipid co-delivery condition. For those of you who are unfamiliar with biological terminology, IC50, or the half maximal inhibitory concentration, refers to the concentration of a drug or substance that is needed to inhibit a given biological process by 50%. In order to calculate the IC50 of a drug through an MTT assay, you need to drug the cells in a concentration gradient and run a regression through the calculated cell viability percentages, which will yield you the percentage at which your compound or substance inhibits the proliferation of 50% of the cells in your sample. The lower the IC50 value, the more potent your compound is. All of the calculated IC50 values are plotted on this heat map, where the higher ones are represented by yellow and the lower values are more purple. Upon analysis, in 4 out of the 6 drugs, there was a trend across all cell lines where co-delivery with sterol, our only fully saturated tripod lipid, led to a reduction in IC50 compared to the drug by itself. This trend is especially prevalent in ammonified, a cationic topoisomerase inhibitor, where the IC50 value when co-delivered with sterol is reduced by up to 99%. However, we can't prove that this trend is statistically significant, and since we had some solubility issues with the sterol lipid, it is possible that the results are skewed. Despite this, we still think it is worth further investigation. Upon seeing this trend, we wanted to see if it was replicable across another colorectal cancer cell line. We ran MTT in the murine colorectal cancer cell line CT26. Upon analysis, it can be seen that all 6 drugs were generally less cytotoxic in CT26 compared to the other 3 cell lines. In addition, we were unable to replicate the lower IC50 trend among sterile co-delivery in the CT26 cell line. We suspect that this is a result of our sterile dilution going bad over time, rather than an indicator that the trend is irreplicable. We think this is the case because we ran this assay a couple weeks before the initial 3 cell lines, and the sterile lipid appeared visibly less soluble in the dilution plate after a few weeks. Sterile itself was our most solid lipid and had solubility issues throughout the initial assays as well. To determine whether the physical properties of our tripod lipids affected the kinetics of drug uptake, we conducted a washout assay on the compound ammonified in the cell line HCT116. We drugged the cell line with ammonified at different concentrations, co-delivered with our 4 lipids. We allowed the cells to sit in drug media for 2 minutes, 5 minutes, and 10-minute time points, where at the end of the time point, the drug media was replaced with fresh media and the cells were allowed to incubate for 96 hours. For the infinite time point, the drug media was not replaced. Standard MTT and LDH procedures were then executed, and the results are displayed here. As you can see, for the washout time points, the longer the cells were exposed to ammonified, the higher the IC50 value. However, the IC50 values at the infinite time point, where the cells were allowed to incubate for 96 hours in the drug media, were lower than the 5 and 10-minute IC50 values. The alladal condition had the highest IC50 value across the 3 time points, and was the only condition where the IC50 was higher than that of the no lipid control. Linoleo had the next highest IC50, followed by oleol and lastly sterol. Once again, the most saturated lipid resulted in the greatest decrease in cell viability.
After we had done the MTT and LDH data, we wanted to run imaging data to gain a better understanding of what is really happening when we co-deliver our lipids with the FDA-approved drugs that Evie showed earlier. Using TBS SN38, doxorubicin, and ammonified that have blue, red, and yellow fluorescence respectively, we hope to see if our lipids help with drug delivery. Although ammonified and doxorubicin didn't really show much data, this might have been due to it not being concentrated enough or fluorescence not being visibly shown, uh, which hindered our ability to quantitatively show how our lipid helps with drug delivery. TBS SN38 showed pretty good data as shown here, although it's not really shown well. It still showed that similar to the washout assay, TBS SN38 saw little to no difference in cell permeability over 5 minutes between the no lipid positive control and the other lipids. Due to this, we wanted to find another way for our drugs to get into cells. We wanted to do this through a CaP lipid complex or a nanothistle. We hoped that this would help our drugs to deliver, our lipids to help deliver drugs through localized delivery. And we made the CaP lipid complex by first using 1 milliliter of pre-made CaP solution that was made by Dr. Yamamoto, centrifuging it down and removing the supernatant, and adding our lipid in chloroform to the solution. And after vortexing and centrifuging and removing the supernatant once more, we had our CaP lipid complex. After finishing formulating the CaP lipid complex, we wanted to evaluate the presence of nanoparticles would decrease cell viability compared to the presence of the tripod lipid. We proceeded to conduct MTT and LDH assays in the HCT116 cell line, except we replaced our tripod lipid with a lipid-CaP complex. And as you can see, the cap lipid resulted in a lower IC50 value across pretty much all of our lipid formulations, which we would like to explore a bit further. Shifting gears, we wanted to perform a fluorescence microscopy to gain a better understanding of whether or not these work. And we saw how ammonified the drugs got into cells pretty randomly, and we couldn't really tell whether or not it was due to the cap locally delivering the drugs into cells or through another pathway. The SN38 also showed the same trend of not really showing much of anything. And one of our hypotheses of what happened would be that when we drug the cells, the cap lipid complex, when we moved it around, might have moved to another location, which would explain why our cells are fluorescing when they're not close to the cap, which is why we want to replicate this data without moving the plate.
So given all the fluorescence imaging that we have done, we now decided to shift gears and explore the potential applications of our tripod lipids in the synthesis of fluorescence quantum dots. So a brief overview of what quantum dots are. They're tiny semiconductor nanocrystals, which means they have properties between insulators and conductors. And quantum dots are really small. They're only a few nanometers wide, and they already have many applications, such as being used in photocatalysis, in batteries, or even in LED lights. So they are able to create all these different lights due to their quantum confinement. Basically, this means that smaller quantum dots emit shorter wavelengths, while larger quantum dots emit longer wavelengths. The image on the bottom left shows how quantum dots absorb and emit light using incoming and pumping beams. So the main circle that you see is the quantum dot, which represents the the small nanocrystal that traps all of these beams. The blue beam is the incoming beam. That represents a photon or light wave that hits the quantum dot, and it excites an electron from the valence band to the conduction band. The pumping beam in purple is another light source, and it helps excite electrons by generating electron-hole pairs, also known as excitons. On the top, electrons are pictured, and the holes are pictured on the bottom. So electrons are negatively charged particles, whereas the holes are positively charged gaps in the valence band, and they interact through Coulomb forces inside a quantum dot. As for the types of interactions denoted by the yellow arrows, there's the VEE, which is electron-electron repulsion. There's hole-hole repulsion. And lastly, there's electron-hole attraction, which is the most important force, as it allows for recombination and light emission. So on the right side, you'll see a light beam coming out. This happens when an electron and hole recombine and a photon is emitted. So now we'll be discussing how exactly we made these quantum dots. So this was actually made possible by previously published literature from the Landry lab in 2014. So we start by preparing two precursor solutions. The first solution is the selenium solution. And as you can see, one of the reactants, trioctophosphine, kind of looks like one of our tripod lipids. So this is the reactant that we are kind of experimenting with and substituting with our sterol, oleo, linoleal, or illetal tripod lipid. The other precursor solution is the cation solution. And one thing to note is that this has to be heated up until 135 degrees Celsius. And while after we complete that step, we can then prepare the growth solution. So essentially, this is just heating up 10 mils of our solvent octadecene to 165 degrees Celsius. In the reaction setup pictured here, you can see that we have a round bottom flask on top of a sand bath, which also has a thermocouple inside. So the setup helps us better monitor the temperature so it's not above or below the temperature, which is important because we have to keep the temperature constant throughout the entire experiment. So after the growth solution is heated enough, then we can add in our precursor solutions. We do this by pipetting 1 ml of the selenium and cadmium solutions at the exact same time. And as soon as these precursor solutions are added into the flask, a selenium and cadmium complex starts forming, so we immediately start the timer. So then at the time points that we have pictured on the table, we withdraw 10 different 1 ml aliquots. And so this experiment typically requires 3 people. We have at least 3 people. We have one person on the timer just counting down right before we have to withdraw an aliquot. One person is doing the actual syringing, and one person is handing the person syringing the different vials. So it's quite an exciting reaction to run. So with these different reaction times, the longer reaction time allows more time for the selenium and cadmium complex to form. So longer reaction time leads to larger quantum dots. And this is how our quantum dots look. As you can see, under normal light, they don't really look that special. It's just kind of clear to a dark yellowish solution. But under long wave UV, they fluoresce, and it's a whole scale from bluish to like a more reddish wavelength. So we're able to quantify that fluorescence using fluorescence spectroscopy. And so there's 2 things to note in this raw data. First of all, the higher wavelength, which are the redder wavelengths, are actually weaker than the blue samples. Our most redshifted vial is actually 13 times weaker than our blue vial in fluorescence. In addition, you can see that we have multiple excitation wavelengths and their doubles, but in analyzation, these are actually just considered as noise. So after some data processing in which we remove that excitation wavelength noise and also normalize all the peaks to the same height, you can see that the graph does become more redshifted as the quantum dot size increases. So this matches up with our expected result. So why do these larger quantum dots emit longer wavelengths of light? To understand this, we kind of turn to a classic quantum mechanics concept, which is the particle in a box model. So in bulk semiconductors, such as silicon, electrons are able to move freely within or between energy bands. So they're like almost continuous. However, in our smaller crystals, such as our quantum dots, which are only a few nanometers wide, things are different. As you have to, according to the particle in the box model, when you confine a particle into a small space with rigid walls, its allowed energy levels become more discrete, which means that it can only exist at specific energy states. So the smaller the box, the larger the energy gaps are between these allowed states. And this makes sense because smaller quantum dots have less atoms and therefore less overlapping of their orbitals or energy levels. And this causes the band gap to become even wider. Same the other way around, if the box is larger, the energy levels get closer together. And if this is hard to visualize for those who prefer like a mathematical equation, on the right, we have the equation for the energy of level n. So our variable L represents the width of the box, which in our specific case represents the size of our quantum dot. And as you can see, smaller quantum dots, which is with a smaller L value, create larger energy spacing, while larger quantum dots have closer energy spacing with a smaller energy value. So knowing how the energy levels differ, we can then relate it to the wavelengths of light emitted. The color of the light depends on the energy of the photon, and that can be determined by the Planck-Einstein relation, which is given in the bottom right corner. Lambda is the wavelength of the photon, and as you can see, energy and wavelength are inversely proportional in this equation. So to sum it up, a smaller quantum dot have energy levels that are even further apart, which means they have higher energy transitions. And then using the Planck-Einstein relation, that means it has smaller wavelengths, and smaller wavelengths correspond to bluer light. So this is why on the really far left of our bio lineups, the ones with this least reaction time will correspond to the bluest fluorescence light.
Okay, so for our future plans, we hope to run MTT and LDH assays on the Calu-1 cell line and redo the CT26 cell line. We also plan to rerun the CaP imaging data so that the plate doesn't move, and formulate quantum dots with sterile, oleal, linoleal, and allyl tripod lipids, as well as maybe put quantum dots into cells.
Okay, so lastly, we'd like to give some acknowledgements. We have other new lab members working on this project, including Elizabeth, Tiffany, and Alina. For many of our pictures on the slide deck, we use BioRender. And of course, we would like to thank ASDRP for giving us all these materials and a laboratory to conduct our experiments. We'd also like to thank our advisor, Dr. Edward New, because he provides a lot of guidance throughout our project. And lastly, we have collaborators such as Dr. Yamamoto, who helped us formulate the CaP lipids, and Dr. Joseph Pazzi's lab, including Dia, Lillian, and Charlotte, as they are working on CMC data for our tripod lipids. Thank you.